Bacterial synthesis of zinc oxide nanoparticles, ZnO NPs, also known as biogenic synthesis or green synthesis, is an eco-friendly and cost-effective method that utilizes microorganisms to produce nanoparticles. This approach leverages the ability of bacteria to tolerate and reduce metal ions into their metallic or metal oxide forms. Here's a general outline of the steps involved, though specific protocols can vary depending on the bacterial strain and desired nanoparticle characteristics. Preparation of bacterial culture. 1. Selection of zinc-tolerant bacteria. Identify and isolate bacterial strains known for their ability to tolerate and reduce zinc ions. Examples include Serratia nematodiphylla, Microbacterium sp., and certain Lactobacillus species. 2. Culture preparation. Grow the selected bacterial strain in a suitable liquid culture medium, e.g., Luria bertani, Lb, broth, nutrient broth, under optimized conditions, temperature, pH, aeration, to obtain a dense cell culture. This typically involves inoculating the broth with a pure bacterial culture, incubating the culture, often at 30 to 37 degrees Celsius, with shaking, e.g., 200 revolutions per minute, for 24 to 48 hours until a desired optical density, OD, is reached. 3. Preparation of cell-free supernatant, for extracellular synthesis. If the synthesis is desired to be extracellular, using biomolecules secreted by the bacteria, the bacterial cells are separated from the culture broth by centrifugation. The supernatant, rich in enzymes, proteins, and other biomolecules, is then used for nanoparticle synthesis. 2. Nanoparticle synthesis. 1. Preparation of zinc precursor solution. Prepare a solution of a zinc salt, e.g., zinc sulfate, zinc nitrate, zinc acetate, in deionized water. The concentration of the zinc salt is a crucial factor influencing nanoparticle size and morphology. 2. Mixing and reaction. For extracellular synthesis, the cell-free bacterial supernatant is mixed with the zinc precursor solution. For intracellular synthesis, the bacterial culture, with cells, is directly exposed to the zinc precursor solution. 3. Incubation and reaction conditions. The mixture is incubated under controlled conditions, temperature, pH, and sometimes agitation. Temperature, often elevated, e.g., 40 to 80 degrees Celsius, to facilitate the reaction. pH. The pH of the solution plays a critical role in the formation and stability of ZnO NPs. An alkaline pH, e.g., 8 to 11, is often preferred for precipitation. Time. The incubation time can vary from several hours to a few days, depending on the desired yield and particle size. Observation. The formation of ZnO NPs is often indicated by a change in color of the solution, e.g., from colorless to whitish, or the appearance of a white precipitate. 3. Purification and isolation of ZnO nanoparticles. 1. Centrifugation. After the reaction, the mixture is centrifuged at high speeds, e.g., 10,000 to 14,000 revolutions per minute, to separate the synthesized nanoparticles from the bacterial cells, if intracellular synthesis occurred, or from the remaining supernatant. 2. Washing. The collected pellet containing the ZnO NPs is washed multiple times with distilled water and/or ethanol to remove any unreacted precursors, media components, or bacterial residues. This helps in purifying the nanoparticles. 3. Drying. The purified nanoparticles are then dried. This can be done by oven drying, e.g., at 80 to 120 degrees Celsius, or freeze drying to obtain a powdered form of ZnO NPs. 4. Calcination, optional. In some protocols, the dried nanoparticles may undergo calcination at a higher temperature, e.g., 500 to 600 degrees Celsius, to improve crystallinity and remove any residual organic matter. IV. Characterization of synthesized ZnO nanoparticles. After synthesis and purification, the biogenic ZnO NPs are characterized using various analytical techniques to confirm their formation and evaluate their properties. UV-vis spectroscopy, to confirm the formation of ZnO NPs by observing their characteristic absorption peak, typically in the UV region, around 350 to 380 nanometers. X-ray diffraction, XRD, to determine the crystalline structure, phase purity, and average crystallite size of the nanoparticles using the Debye-Scherer formula. transmission electron microscopy, TEM, and scanning electron microscopy, SEM, to visualize the morphology, shape, e.g., spherical, rod-like, hexagonal, and size distribution of the nanoparticles. Energy dispersive X-ray spectroscopy, EDX, EDS, to confirm the elemental composition of the nanoparticles, presence of zinc and oxygen. Fourier transform infrared, FTIR, spectroscopy, to identify functional groups from biomolecules, proteins, enzymes, polysaccharides, that may be capping or stabilizing the nanoparticles. Dynamic light scattering, DLS, and zeta potential, to determine the hydrodynamic size, polydispersity index, and surface charge of the nanoparticles, which are important for their stability in solution. Other techniques: high-performance liquid chromatography, HPLC, Raman spectroscopy, photoluminescence, PL, and BET surface area analysis can also be used for further characterization.